Hey everyone, welcome to The Explainer. Let's just dive right into this, shall we? If you are prepping for the Cambridge AS and A-Level Biology 9700 exams, well, you probably already know that memorizing the textbook inside and out, that's really only half the battle. Because the difference between scoring a B and landing that elusive A-star often comes down to something that isn't even explicitly taught in the syllabus. It comes down to understanding the examiner's secret framework. And today, we're going to crack that code. We are looking at the visual playbook that literally separates the top-tier candidates from everyone else.
Here is our roadmap for this explainer. First, the high-mark exam strategy. Second, decoding complex biological data. Third, avoiding the generic answer trap. Fourth, the method mark lifeline. And finally, number 5, examiner red flags revealed.
All right, Section 1, the high mark exam strategy. Now, I really want you to ask yourself something. Why is it that so many incredibly smart, super well-prepared students find interpreting complex data and graphs so ridiculously hard under exam conditions? I mean, they know the biology, right? But when they're faced with a massive data table or a complex chart, their marks just sort of slip away. Why is that? Well, it's actually because they're missing the core framework that examiners actually use to award points.
Check out this complete breakdown of that exact framework. Notice that ACE strategy right there at the top left? That is your golden ticket. It stands for concept, evidence, reasoning, and wording. Basically, every top band answer starts with the core biological concept, backs it up with data-driven evidence straight from the stimulus, applies solid scientific reasoning, and then finishes off with specific examiner-approved wording. You can see how this perfectly applies to case studies like enzymes, right? Whether you're discussing the lock and key or the induced fit hypotheses, examiners are prioritizing this connected, conceptual understanding way over just isolated facts.
Let's break down how this actually builds into an actionable sequence for you. Step 1, identify the concept. Step 2, extract specific evidence from whatever graphs or tables you're given. Step 3, apply logical scientific reasoning to connect that data back to the theory. And step 4, use precise examiner wording based on the command words. If you structure every single long-form answer using these 4 steps, you are aligning perfectly with the examiner's mark scheme.
Moving on to Section 2, decoding complex biological data.
Before we dive into our next visual, just look at this comparison. A standard, flawed student answer usually confuses cause and effect, or it just blindly describes a trend without any true analysis. But a top-band answer? It uses comparative connectives. It explicitly points out similarities and differences. It doesn't just say, this went up and that went down. It logically connects those movements.
And this brilliantly illustrates exactly why that level of detail is so critical. Topic 5.1, the mitotic cell cycle, frequently determines grade boundaries. In fact, it often makes up around 15% of Paper 2's total marks. And honestly, losing up to 15% of your grade to a single, easily avoidable mistake is a pretty tough pill to swallow. So, to secure these marks, you absolutely have to deconstruct the cell cycle using specific procedural workflows. You can clearly see the examiner's warning zone laid out here. Do not confuse cause and effect. You've got to handle the information systematically, and crucially, you need to know exactly how to select the right statistical tools when you're comparing different cell stages.
Let's quickly distill those statistical tests from the diagram into a super simple, memorable checklist you can use when you're extracting data. If you're comparing means, you use the t-test. If you're dealing with categorical data, it's chi-squared. And if you're looking for relationships between variables, you're going to use a correlation test. Just lock this checklist in your head because choosing the wrong test is a massive missed opportunity for some seriously easy marks.
Let's check out another high-yield area: energy values of carbohydrates, lipids, and proteins. Choosing the right statistical test here, like we see in the methodology section, prevents you from falling into what examiners call the vague answer trap. Because top candidates don't just quote numbers, they link energy values to other areas of the syllabus, like molecular structure or aerobic respiration. And that cross-topic advantage, that's exactly the kind of thing that pushes a student from an A to an A-star.
All right, Section 3, avoiding the generic answer trap.
So the really crucial point here is understanding what examiners absolutely hate. Take a look at this flawed answer. The pigments separated because they are different. Ouch. That is a classic red pen moment, right? It's completely superficial. Now compare that to the top band requirement. It demands that you use precise command words, uncover anomalies in the data, and actually extract specific contextual information instead of just throwing out broad, generic statements.
Let's see this in action with chromatography.
The painful learner mistake is right there, front and center. Examiners despise generic answers. When it comes to chromatography, analytical depth means you're extracting specific data, calculating statistics, and drawing evidence-based conclusions. You can't just describe the trends, you have to explain the why, using the proper scientific terms. Basically, you have to demonstrate a logical, structured, and connected understanding.
And we see this exact same vagueness trap absolutely destroy marks when students are asked about water as a solvent in blood or its high specific heat capacity. Notice those mark killer mistakes highlighted there? A student might give a generalized definition, but then completely fail to link any specific data, or they just totally forget to include their units. To unlock those top-band marks, you have to deconstruct the provided information, solve it through a systematic, step-by-step process, and always evaluate your answer to see if it's reasonable. Bottom line, always apply the data and use the correct terminology.
Which brings us to Section 4, the method mark lifeline.
Let me ask you something that might cause a little bit of anxiety. What happens if you are under immense time pressure? You're calculating an energy yield or a water potential gradient, and your final numerical answer on the exam is just completely, utterly wrong. Is all hope completely lost for that question?
Well, let's look at the energy yield in aerobic versus anaerobic respiration to find out. Right there in the middle, we see the absolute beauty of the method mark strategy, because examiners actually reward the analytical process itself. Extracting data from graphs and showing clear working is so essential, because Cambridge examiners will actually award you method marks for using the correct process, even if your final numerical result is totally incorrect, just because of a silly calculator error.
I honestly cannot emphasize this enough. Always show you're working. Write down every single step of your math. Method marks can literally save your grade and bump you across a boundary, even if your conclusion is totally off. Just never leave your calculations blank, and never, ever just write down the final number without showing how you actually got there.
Finally, Section 5, Examiner Red Flags Revealed.
I want you to read this authoritative quote straight from a principal examiner report. Students confuse cause and effect, forget to check units and terminology, and provide answers not specific to the context. Look, this isn't just friendly advice. This is undeniable, real-world examiner feedback telling us exactly where students are bleeding marks, year after year. When we look at STO model responses, we can pinpoint these exact red flags right there on the right-hand side. The generic answer trap rears its ugly head again when students just provide a memorized textbook answer instead of actually applying it to the specific environmental scenario they were given. Then you've got the terminology mismatch, like confusing water potential with concentration gradients in stomatal cells. And finally, unit negligence, literally just forgetting to check the units on the stimulus tables.
We'll wrap up our visual case studies by seeing how these fatal flaws manifest in topic 15.2, looking at gibberellin pathways in barley. Check out the red pen annotations here. Confusing cause and effect in these hormonal pathways is a massive, top examiner reported mistake. The real key to getting that evaluation edge is comparing the different approaches, evaluating alternative interpretations, and making absolutely sure your answers are firmly rooted in the specific data context of the question. All right, let's recap this Highmark framework into 4 actionable takeaways that you can literally use in your very next study session. Number 1, always deconstruct the question and extract your specific data first. Number 2, employ that ACE framework we talked about: concept, evidence, reasoning, and wording. Number 3, always show clear working so you can secure those method marks. And number 4, rigorously avoid generic answers by grounding absolutely everything in specific context and correct units. So, you now possess the examiner's secret playbook. You know the strategies, you know the pitfalls, and you have the exact visual blueprints to secure those top marks. The only question left is, how are you going to actively apply this framework to decode your next past paper? Honestly, don't just passively read your textbook anymore. Grab a past paper, apply the ACE framework, show you're working, and start thinking exactly like an examiner. Thanks so much for joining me for this explainer, and happy studying!